This story is not just one of two men in a Eureka moment, but a complex tapestry of collaboration, competition, inspiration, and, unfortunately, controversy. In the early 1950s, a fundamental question in biology remained unanswered. How is genetic information stored and transmitted from one generation to the next? It was known that DNA, deoxyribonucleic acid, was a key player, but its structure and how it functioned were a complete mystery. Unlocking its structure was seen as the key to understanding the mechanism of life itself. Several world-class research groups were in a quiet but intense race to find the answer. James Watson, American. A young, brilliant, and intensely ambitious zoologist who became obsessed with genetics after hearing about the role of DNA. Francis Crick, British. A talkative, boisterous, and brilliant physicist who had moved into biology. He possessed a formidable talent for theoretical model building and X-ray crystallography theory. Maurice Wilkins, New Zealand-born British. A quiet, cautious physicist at King's College London, working on DNA using X-ray crystallography. He saw DNA as a physics problem. Rosalind Franklin, British. An expert X-ray crystallographer at King's College London working under the direction of John Randall. She was meticulous, sharp, and produced the best X-ray diffraction images of DNA in the world. Her relationship with Wilkins was strained and full of miscommunication. They worked on the same subject, but not as a team. Linus Pauling, American. The world's leading chemist at Caltech who had already discovered the alpha helix structure of proteins. He was the favorite to win the race. Watson and Crick met at the Cavendish Laboratory at the University of Cambridge, which was headed by Sir Lawrence Bragg, a Nobel laureate in physics himself. They were not officially supposed to be working on DNA. Their assigned project was the structure of proteins. Their interest in DNA was a side project, pursued with Bragg's reluctant tolerance. The misfire and the shutdown, 1951-52. Watson attended a seminar by Rosalind Franklin where she presented her data. He, lacking a strong background in crystallography, misunderstood key aspects of her talk, notably the water content of the DNA samples. Based on this flawed understanding, he and Crick built a three-chain model with the phosphate groups on the inside. They excitedly invited the King's College group to see it. Franklin immediately pointed out the errors. Her data clearly showed that DNA had the phosphate groups on the outside and required much more water than their model allowed. The model was embarrassingly wrong. The failure was so severe that Sir Lawrence Bragg ordered them to abandon DNA research and leave it to the King's team. They returned to their work on proteins. The Photo 51 and the Pauling threat, late 1952, early 1953. Despite the order, Watson and Crick continued to think about DNA. The race reignited when they learned that Linus Pauling had proposed a triple helix model for DNA. Upon seeing his paper, they immediately spotted the same fatal flaw that had doomed their first model. Pauling had placed the phosphate groups on the inside. They knew he was wrong, but they also knew the greatest chemist in the world would quickly realize his error and come back with the correct solution. The pressure was immense. Crucially, in January 1953, Maurice Wilkins, frustrated by his lack of progress with Franklin, showed Watson Rosalind Franklin's finest X-ray diffraction image, Photo 51, without her knowledge or consent. The moment Watson saw the photo, he later wrote, "My mouth fell open and my pulse began to race." The striking, clear X-shaped pattern was the unmistakable signature of a helix. More than that, the pattern suggested specific parameters like the pitch and the diameter of the helix. This image was a vital clue, the Chargaff rules in the final pieces. Around the same time, Crick was shown a report containing Franklin's data from a medical research council meeting. This data included her precise measurements, which further confirmed the helical structure and its dimensions. Separately, the work of biochemist Erwin Chargaff became crucial. Chargaff had discovered that in DNA, the amount of adenine, A, always equals thymine, T, and the amount of guanine, G, always equals cytosine, C. This was known as Chargaff's rules, but no one had yet understood their structural significance. Watson and Crick now had all the key pieces. 1. Franklin's data confirmed a helix, 2 chains, phosphate backbone on the outside, and the precise measurements. 2. Photo 51 provided a visual smoking gun for the helical structure. Chargaff's rules, ART and G.C. The Eureka moment. The double helix, February-March 1953. With the pieces in place, Watson began trying to build physical models, like Tinker Toys, of the nucleotides to see how they might fit together. His first attempt was pairing like with like, A with A, etc., which didn't work. Then on February 28, 1953, as he was shuffling cardboard cutouts of the bases on his desk, he had a breakthrough. He realized that an adenine base, a purine, paired with thymine, a pyrimidine, formed a shape that exactly matched the shape of a guanine-cytosine pair. This was the final crucial insight. Not only did the pairs fit together perfectly held by hydrogen bonds, but it also explained Chargaff's rules, A always paired with T and G always paired with C. This meant the two strands were complementary. If you separated them, each could act as a template to recreate the other, explaining how genetic information could be copied. They built the full model, two chains twisting in a right-handed double helix with the sugar phosphate backbones on the outside and the paired bases forming the steps on the inside. Everything fit Franklin's data perfectly. They showed the model to Wilkins and Franklin, who immediately saw it was correct. Franklin's famous reaction was one of scientific integrity. Her comment was that the model, while built with her data, was consistent with all the evidence she had. On April 25, 1953, Watson and Crick published their seminal paper in the journal Nature, titled Molecular Structure of Nucleic Acids, a Structure for Deoxyribose Nucleic Acid. It was a mere 900 words long. It contained no new experimental data. It was a theoretical model built almost entirely on the data of others. It was accompanied by papers from Wilkins and Franklin, which provided the critical experimental evidence supporting their model. The paper's famous understated conclusion: it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. The aftermath, controversy, and legacy, the Nobel Prize. In 1962, James Watson, Francis Crick, and Maurice Wilkins were awarded the Nobel Prize in Physiology or Medicine for their discovery. The Nobel rules at the time stipulated a maximum of 3 winners and did not award prizes posthumously. The Rosalind Franklin controversy. Rosalind Franklin had died of ovarian cancer in 1958 at the age of 37. The Nobel Prize cannot be awarded posthumously, so she was never considered. The controversy stems from how her data was used. Watson and Crick did not steal her data in the legal sense. They were shown it by a colleague, Wilkins. However, they used her unpublished precise data and her photograph without her knowledge or permission to build their model. They did not adequately acknowledge her crucial contribution in their initial papers. Many historians of science believe that had Franklin lived, the Nobel Committee might have faced an impossible choice, potentially leading to her being recognized alongside Wilkins. Franklin was not a victim waiting to be saved. She was an exceptional scientist who was closing in on the structure herself. Evidence suggests she was moving away from the idea of a helix for a time, but was very close to putting all the pieces together. Watson and Crick's genius was in model building, theoretical insight, and synthesizing the information from multiple sources with incredible speed. The legacy. The discovery of the double helix is one of the most important scientific achievements of the 20th century. It launched the modern era of molecular biology and genetics. The structure itself revealed the secret of life. Replication. The complementary base pairing showed how DNA could be precisely copied. Information storage. The sequence of bases along the helix encoded genetic information. It directly led to the cracking of the genetic code, the development of recombinant DNA technology, the Human Genome Project, and all the advances in genetics and medicine that we see today. It was a discovery that truly changed the world.